Good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Pharmaceutical Case Studies Illustrating Data Analytics Strategies, Extracting the Most Value from Your Data Assets. My name is Diane and I will be your X-Talks host for today. And today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. And this webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit any questions or comments you may have for our speakers throughout the presentation, and you may do so by using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen, and if you require any other assistance, please contact me at any time by sending a message using this chat panel. At this time, all participants are in a listen-only mode, and please note that this event will be recorded and made available for future download. At this point, I would like to thank Alchemy Innovation and Seeq, who developed the content for this webinar. Alchemy Innovation Inc. is an engineering company providing technical and business support for data-driven pharmaceutical process development. The company is founded on the premise that companies need better science-based technology solutions that provide specific technical direction and guidance from the extensive data. Alchemy Innovation also partners with software companies who want to tailor their hardware and software solutions to the scientists and engineers who run the R&D, pilot and manufacturing scale processes in pharmaceuticals and biopharmaceuticals. Seeq is a venture-backed software company with headquarters in Seattle, Washington. The company is founded on the premise that process manufacturing companies need better solutions for deriving business insight from industrial process data. The Seeq decision support environment features a HTML-based client application and server-based platform, is the first modern application designed specifically for accelerating insight into time series data. And Seeq has a successful and accomplished executive team which brings over 100 years of experience and expertise in process manufacturing, mission-critical systems, and software development from OCSoft, Microsoft, Boeing, and Honeywell. And now I would like to introduce our speakers for today's webinar. Our first speaker for today is Dr. Lisa Graham. Lisa is a registered professional chemical engineer with 20 years technical and business experience focused on pharmaceuticals, life sciences, and specialty chemicals. She has a strong business background through senior leadership positions. Technical strengths include chemical engineering expertise, data analytics, applied mathematics, process model development, product development expertise, and process analytical technology applications. And she holds a Bachelor of Science degree and PhD degree in chemical engineering from Oregon State University. She is the author of 13 scientific papers and 25 professional presentations with 2 patents. And now I would like to introduce our second speaker for today's webinar, Brian Crandall. Brian has 16 years experience in process automation in a variety of roles, each with an emphasis in biotech and pharma. As a project engineer, he architected batch control strategies for several large-scale commercial production facilities. During his software engineering role, he developed applications to analyze batch programs and historical process data. As a product marketing manager, he focused on data integration between the control system and business platforms to improve manufacturing efficiency, and his current passion is providing data analytics to accelerate production and empower operators, engineers, and data scientists. And he holds a MS degree in software engineering from the University of Texas and a BS degree in engineering from the Colorado School of Mines. And now I would like to hand over the mic to our first speaker for today, Dr. Lisa Graham. And Lisa, you may begin when you're ready.
Great, thank you very much. I appreciate that introduction. So I see that the slides are being put into presentation mode. Thanks, Brian. All right, so hello. Thank you everyone for joining us today. To get started on the next slide, I want to draw your attention to the cartoon on the left. So as you can see, it says, let's shrink big data into small data and hope it magically becomes great data. Humorous, maybe for a Monday, but we all recognize that hope is not a strategy. So today we're going to focus on some real steps that we can all take to truly extract the most value from our data. As part of our presentation, we plan to step through key elements of a data analytics framework that give you some concrete tools and ideas for how to make changes for what you do, but as well as what you can do for the organization that you're in. As part of this, we plan to use a series of case studies that help illustrate how to approach this and perhaps build confidence with the fact that a lot of what we're showing today is in real practice with some of your colleagues and other companies. So to begin, let's start by reviewing our current reality on the next slide. So what is our current reality? Our current reality is a world in which we've created some amazing measurement technology. We've built some incredible processes, we've grown pipelines of new medicines, and all, frankly, with a pretty lean workforce. It's probably about once a week I hear folks say, I've got to do more with less. We have so many promising therapies, how do we get them through? Well, as a result of all of this advancement in technology, we have tons of operational data, whether it's the R&D scale, the pilot, or commercial scale. And it's not just technical, there's business system information as well. And however, the unfortunate side effect of generating this much data, particularly where we haven't had necessarily the right tools or applications for having a clear data analytics strategy, has resulted in the kinds of struggles that we have illustrated here in each of these blue boxes. If you're joining us today, then we believe you're one of the people that are willing to acknowledge that these struggles exist and are open-minded about rethinking how these things are done today. Let's eliminate the phrase, it's faster to do the experiments again. It shouldn't be faster to do them again. It should be faster to find what we did. So what's what is at the core of this challenge? Well, it's simply the fact that our data is not well identified or not well connected, and we find ourselves, frankly, not knowing where to look for the data. In many cases, we don't even know what data exists to be looked for, and that's particularly true as people move in and out of organizations or in or out of groups. Or if you look ahead in time in your organization, my guess is you have some very senior people that will be retiring. And we all know that we can go to them for that knowledge that's in their head. So as a result of the situation, we end up with an iterative data processing process as shown on the next slide. And how are we all managing this? Pretty much through, I would imagine, each one of you has a different situation here, but what I've seen through some of my work is overly constrained databases that make it hard to add, let's say, new analytical technology data. Excel spreadsheets that are typically the result of some time-consuming effort. That Excel file is then stuck somewhere electronically, not accessible to others. And then, and how is this reality impacting the people side of our organizations? Well, right at the heart of being effective with their time and energy, as we see on the next slide. The challenge of working with the process data these days is that our scientists and engineers spend a significant amount of time, frankly, messing around with the data and less time using that information to solve problems. These are very talented people, and while tools like Excel are useful, there are better ways to access and contextualize that information and collaborate. So enough about the problem. I'm guessing this is frankly not news to you because you're here with us today. So let's get into the solution. And we aren't talking about a solution that says something magical is going to happen if we keep doing what we're doing. I often hear we're too busy and we'll make things work, but we need to actually take steps, as you know. So let's move on to the solution by first defining the data analytics framework and the key elements. And so what is the first key element of our strategy? Well, it's knowing exactly what we want to achieve. So as shown here, in some cases, the data analytics strategy needs to deliver a functional relationship between the product quality attributes and the related chemistry or biology in the processing environment, whether it's in the API process in small molecules or the bioreactor in large molecules. This might include coupling optimized process analytical technology solutions with a comprehensive data analytics application. In other situations, there are a number of other business drivers that our case studies will illustrate in a few minutes. Knowing the big picture is important from a scientific standpoint, as shown on this slide. But as we show on the next slide, so is having a strong grasp on the day-to-day questions that each person in the organization needs answered. This is an equally important element in designing the data analytics framework, both what we're trying to accomplish and who is trying to accomplish it. A few example roles are shown here. I'm hoping you identify with somebody in this particular diagram. I mean, every company is different in how it's organized. So take it as a placeholder to illustrate. There might be the engineer who's working with clinical or manufacturing team. And what keeps them up at night is, how am I going to answer questions about the data to support a filing or the FDA query that just came in? Or the senior scientist in your organization who's responsible for trying to guide portfolio development by being able to access information across a multitude of experimental findings, not just one experiment or one product. Not to mention our lab scientists who wants to be as efficient as possible in their analysis and engineering design to advance the very specific product that they're responsible for. So let's move from the problem statement to a solution that can be used to support all of these roles in the next slide. So first, we have our data sources shown here on the right. And then we have tools available that we use to look at data once we've pulled it together. So in many cases, what's in the middle typically is an Excel spreadsheet. Today's talk focuses on replacing that with an agile data analytics framework where we want all of this disparate data sources to be linked in a way so that it's in one place. And I'm not saying we move the data at all. In fact, I'm saying we leave the data where it is. And we'll talk about that in a little bit. But we simply apply this agile link to all of that data, all of that process data and contextual data, and essentially stitch it together where we have the opportunity to use it with any of our tools we have available. And this is where we leverage the Seeq application. So let's take a closer look at how all this fits together to give us our data analytics framework in the next slide. We'll get into exactly how Seeq works in a few minutes. But first, let's look at what the data analytics framework looks like with all of the key elements included. In this illustration, our data sources exist wherever we store them, and Seeq server connects to wherever that data is. Ultimately, why is this important? Well, it saves us from having an overly constrained database that can't flex with how a company's process analytical technology strategy is naturally going to evolve. And from here, we have access to our Seeq workbench, which is our friendly interface for performing investigations and ultimately getting that important direction out of our data. And finally, we can use the same Seeq workbench for reducing the data sets into information that we can add into the other applications for additional analysis, or to simply share with colleagues, or to have available on key reports. So perhaps it's most helpful at this point to illustrate this approach with an example. So to do that, on the next slide, we're going to focus on a typical process view. So as shown here, whether we're discussing brewing or bioproduction, and believe it or not, they have a lot of similarities when you get down to the science and the kinds of measurements that we take. There are a number of data sets in both cases that are related to the online equipment, whether it's the pressure drop from the pump, across the pumps, the thermocouples, flow meters measuring delivery of liquid. We might even have online probes measuring cell viability. Some of you might use things like the Futura system, for example, to get real-time viable cell concentration. But not only do we have these online data sets, hopefully they're being recorded in a historian like the Delta V or Pi systems, and we'll show you one from a Delta V system in a minute. But we also have at-line or offline data sets from a variety of analytical devices. So to have the full picture, we want to have all of these data assets available for the analytics. So let's begin our first case study on the next slide, where we're going to walk through a real problem statement focused on bioproduction, outline the approach, and show you the results of using the framework. Okay, so diving in, we're going to get into some real science and real data now. This is a recent case study published with Merrimack Pharmaceutical, and it's focused on the challenge identified at the 1,000-liter scale. So imagine you've gone through a whole series of development and other efforts, and you're at the 1,000-liter, and things look like they're going just fine, and your downstream group says, uh-oh, we have an issue, and it's impacting yield, and it puts a clinical trial at potential risk. So what we shared was that we needed an effective data strategy, not only to evaluate what was happening at the 1,000-liter, but also to very quickly be able to look at data from the 100-liter and the 1.5-liter and the upstream process train with high throughput screening. What's the most effective way to iterate and look at what's scientifically driving this? So moving on to the next slide, I just wanted to make sure that we shared a little bit of data that goes along with this. So the yellow and orange and green curves represent the optimized 100-liter and the 1,000-liter GMP batch. So as you can see, from an upstream analysis perspective, things looked very promising. And in fact, it was a point of success at this point for what was being done upstream. However, if we investigate further, we had the downstream group doing sort of the more standard chromatography identified in this next graph on the right a series of curves that were related to an impurity that was happening in such a way that yield was reduced. So if you were in this situation, you have two options for how to approach the data analytics that's going to happen in order to get this figured out, reassessed, and ready within, in this case, a 4-month window before the next opening to do a clinical batch. So the team did a series of experiments that Tara Barrera shared actually at a conference in December, where they narrowed in on the scientific hypothesis of what was happening, did a series of experiments, and then used the framework, as I'll step through in a minute, to be able to assess that quickly and eliminate the need to do all of this in Excel. So going on to the next slide, just to drill down a little bit further, I've mentioned that we have to integrate a number of disparate, separated data sets. So the key variables here for this particular physical situation are the cell health, the cell environment, and the performance. So not only do we want to track individual variables and compare these across scales, we want to look at the relationships between the metabolites and the viable cell density. We want to look at the relationships between titer and metabolites. We don't want to just be restricted to looking at a single variable from that single analytical instrument. We also want to look at these variables in context of process variables like oxygen flow rate. So in addition to all the existing data leading up to these experiments, consider the generation of data in just 6 reactors with multiple sample points per reactor with multiple analytical devices. And it's all this contextual information. So it is a lot of data, and it's an asset that's been created. So on the next slide, I'm going to walk through how we used our framework. So as shown here, we're going to illustrate how to rapidly create cell-specific rate calculations across multiple batches, all in the same analytics environment. So beginning with the time series data related to the oxygen flow rate, we can pull that into our Seeq workbench. And by pull, I essentially mean reference where it's sitting, but be able to see it here. This data, in fact, was being stored in a Delta V data historian. So as you can see here, we pull in or see the 3 3-layer bioreactors, all being run with different hypotheses of what might be happening, and the 100-liter sub for reference. So then we can pull in our offline vi-cell data. So this is our cell viability. And we want to easily overlay that viable cell density and use the environment to calculate an integrated viable cell density, which you can see there in the light curves. And then last, it's pretty easy to do some straightforward math, and in this case, create the cell-specific oxygen uptake rate as a function of equipment scale and conditions. So imagine you could now pull in the 1,000-liter data and relevant screening data from a colleague who captured some information a few weeks ago, and begin asking questions about the scientific fundamentals that work here. I mean, as you can see in the bottom curve, the graph for the 100-liter sub is a very different shape from the 3 liters. So in some respects, it maybe isn't a surprise that they have some different results at this scale. And this isn't just constrained to the bioreactor. So as shown on the next slide, we can do the analyses we showed here, but we can bring in not just the upstream information, but we can also bring in the downstream purification train and begin to correlate performance across both. Because remember, early indication of upstream was that the VCD was looking just fine. However, as we start to look at the downstream, so imagine the ability to use this to bring teams together and collaborate on what's really happening here. It's a bit of a more holistic molecule view. So this is just the first example of today where we can use this framework to easily analyze across different data sets, enable new queries, typically not asked because it's been too hard until now to see all the relevant information, and open a window for cross-group communication and problem solving. And so with that, I'd like to turn this over to Brian, who's going to walk through a number of other examples for us.
Thanks, Lisa. Hello, this is Brian Crandall, and I'm an analytics engineer with Seeq. My role is to work with customers to make them successful with the Seeq application and collaborate on use cases. I also have the pleasure of doing webinars like this to share analytics solutions that we've completed with my partner, Lisa at Alchemy Innovation, and our customers. So I'm looking forward to this. On that topic, Lisa just shared our first use case from Merrimack Pharmaceutical that illustrated a business driver for the integrated upstream and downstream analysis. And we have a number of others listed here. So for those of you joining us today, think about what you personally do day to day and pick out a few of these other business drivers that might be of interest to you. And as I go through these examples, think about how we can use what we show in your day-to-day tasks. And before we jump into how we accomplish these, as Lisa was saying early on, we need to explain how Seeq actually works and where it fits into your data framework. So let me do that. Basically, before we talk about specifics and you see it in action, time series data is hard, and that's just a fact. It's hard to work with for many reasons, and we know this intuitively from trying to do calculations and spreadsheets and custom database queries. Here I distilled it into 3 parts. Number 1 is that the data is continuous throughout all time. There is no clean break in time. Philosophically, you can go backward and forward into infinity, but in practice that usually means when you're working with your data is you're trying to line up timestamps that happen at this point in time with different timestamps that happen in a different point in time on these different signals. And number 2 is that there is a lot of data. Not only do you have years of data in your files and historians, but there are thousands of sensors, so it grows geometrically, just the amount of data. And number 3, there is no context by itself in time series data. It's hard to specify time periods of interest. Do you do it per experiment or do you do it per asset? And how do you navigate it? And how do you combine contexts together to get the answer you want? So our approach, and you'll see this in action soon, is to provide a means to put boundaries around the specific time periods of interest for the engineer and scientist. And we do this in an ad hoc fashion, allowing users to follow their train of thought. And we call these time periods, you can see them on the slide, those horizontal bars, we call those capsules. And capsules can be defined in limitless ways based on time, like a particular day of the week, certain lengths of time, financial quarters, whatever. You can define limits of a signal above or below a certain value, or you can define patterns in your signals, like certain peaks or waveforms. Once we have these capsules, we have successfully wrangled the data. You hear us talk about wrangling data, and this is what we mean when we say wrangle it. Users can filter out all the noise and get the insight they need by only focusing on these time periods that are of interest. So all of the unimportant data of when the machine is off or when there's been no data collected, all that gets filtered out and all those distractions are gone. So the user can just start getting insight. It'll just start jumping off the screen to the engineer and scientist. Once the capsules are defined, the user can apply their domain knowledge to aggregate, collaborate, report, and monitor the data however they want. This could be over a series of experiments, batches, predicting equipment performance, monitoring assets for a specific event to occur, a lot of variety of ways. So let me show you how we apply these concepts to several use cases in the pharma industry. The first use case is to minimize waste in a fill-finish type of process. So let's follow the star and dig in here. This use case was presented by Abbott Nutrition at the OSI Soft Europe conference last year. As they shared, the goal was to optimize the weight control process during packaging to minimize waste. When overfilling occurs, it leads to scrap or rework of the product, and this seemed like a good opportunity to cut cost in their process. Abbott began the process of getting insight by doing calculations in spreadsheets, very common. They did things like loss per fill, variability in the control loop, and then they were able to aggregate this by product and shift that performed it. However, there was a lot of time spent by engineers doing these calculations in spreadsheets. So to save time of the engineers and increase the turnaround time of the analysis, Abbott used Seeq to do these calculations. Their presentation is publicly available on the OSI Soft website, and we've included a link here. And let's show this approach using some example data, and I'll explain how to apply a data analytics framework to do this. So let's start off by opening the Minimize Waste workbook by double-clicking on it. And the first view that you see is a recent feature of Seeq called histograms. One of the metrics Abbott was tracking was a friendly competition between shifts to see who had the lowest overfill. Since this feature wasn't available in Seeq during their presentation last year, I took some artistic license to show how that would look to create an interesting report. Again, this has been mocked up data in our example, but the process is similar. So to go through this analytics process and step through it, I will use another feature of Seeq called the journal. And the journal, let me move over here, is shown on the left. And this is what allows a user to clearly document their thought process and share their analysis step by step, just as I'll do here. So step one, let's start with a blank worksheet. You can see that you're getting prompted to add data. So let's add data. Let's add the filler state and the weight controller set point and process variable to the display. You can see that the top signal indicates the mode of operation. In step three, we created a set of capsules based on shift times for day, night, and swing shifts. And you can click through them to show them and highlight them in the display window. And let me adjust the screen here to get more real estate so we can expand the graph. And then next in step four, I added a search using value search to show when the filler state was in filling mode. So you can see the text filling shows up there. And you can see that those capsules that were created correspond only to those parts of the filler state tag that read filling there. They're shown at the top there. And these capsules are the part of the process that we care about where we want to calculate the overfill of the weight controller. We don't care about the state of the controller unless it's actually filling. So with that, we can move on to the next step, step number 5. I created a calculation to show the difference between the process variable and the set point, and only show the value that was positive, so when there was actually overfilling occurring. So down there at the bottom, you get a quick visual of that as it occurs during that filling mode. And lastly, in step 6, I used a histogram to totalize the overfill by shift. So the histograms allow you to do aggregations in very flexible ways. And here you can see that the swing shift has more overfill than the day shift, while the night shift has none. Again, this is not real data. No offense to anybody, it's not real. But this is what it would look like in a hypothetical situation. And notice that this calculation is automatically being performed over time. So if you want to adjust that time range, you can do it just by going down to that investigate range like I'm doing here, sliding it around. Let me do the last 7 days and watch it update here. So the histogram updates and the calculation is automatically performed. So we'll allow you to go through your time range and get the aggregations however you want. And in the journal, since we have these links in here, I can click on and document a link that shows some other interesting data. So let me click on that. And here I brought up another time period where it shows different results when the day shift had more overfill than the swing shift. So in summary, Abbott Nutrition followed a similar process to what I just showed here to get more visibility into their filling operation. They shared the results with the shifts and all the while saved precious engineering time rather than doing these calculations in spreadsheets. So just to recap, the capsules concept that I introduced earlier, in the previous use case, we created these time slices based on personnel data for shifts and process data for the filling mode. And then within the Seeq application, you can do just like I did. You can combine them, and you saw how seamlessly you can combine them and do aggregations on them. But you can also add other data as well, like maintenance data or shutdowns and work orders. There's really no strict definition to how you define capsules. It's only defined by whatever time periods that you find useful. So the list of context could be a variety of things, continues on, health, safety, environmental, lab, reliability, and inventory data. Okay, so that was the first one. Let's move on to the next use case for improved product quality. And an example of this use case was presented by Bristol Myers Squibb at the OSI Soft conference in San Francisco last March. In their presentation, BMS's goal was to automate a calculation used to check the quality of their chromatography column bed packing. In this case, the symmetry, or asymmetry as it were, of the peak in the conductivity signal indicates the quality of the packing, which can impact the end product quality. So it's an important metric for their production and business. An interesting anecdote of this use case is that the scientists were using paper printouts to do this calculation. They had not even been able to digitize this effort, or else I'm sure they would have. The reason is that working in a spreadsheet didn't offer any benefit because the calculation was too onerous and involved manual eyeballing at the places that the lines intersect. And BMS was able to skip this spreadsheet process and go directly from paper to a modern software application for data analytics. So this is one more paperless solution for the pharma world. And the result was a visual automatic calculation for quality control of the beds and allowed the scientists to spend their time running their experiments instead of doing these calculations. So let's take a look. And by the way, their presentation is publicly available on the OSI Soft website that I showed in the previous slide, and that link is there. So let's show you how it was done. So first off, we'll open the Improved Product Quality workbook by double-clicking on it. And here is the result of the analysis. Each peak is shown with a symmetry calculation in the form of a bar chart, the bottom lane. And my example data is showing some interesting results, so pretty asymmetrical. But let's see the process used to get us to this point. So step one, I like to start with a blank worksheet just to show how easy it is to build up. So click there, so blank worksheet. And next, let's add a UV signal to the trend. I used UV rather than conductivity, but the math will be the same. In step three, actually before we get to step three, this is what the signal looks like. We want to calculate the symmetry of those curves. So in step three, I just did a quick search to find those peaks. So here in this case, I just looked for when that signal was greater than 0. So very easy to do. We have a capsule to visualize it. In step 4, I created a baseline average of the UV prior to the peak. So this is what's used to normalize the different peaks. So I'm getting the average out here, out in front of the peak, and then I plot it right here, right when the peak occurred. It's basically 0 at this point. And next, I created a calculation to define the 10% value, which is the value that is 10% of the range between the maximum peak value and the minimum and the baseline average. And let's put all of this on one axis and on the same lane so we get a better visual of how our analysis is progressing. This is where we can begin to see why paper printouts were used in the past, because they want to do the symmetry at the intersection of those lines. And it's just hard to do in a spreadsheet, actually harder than just seeing it on a piece of paper. So next, I created a set of capsules or conditions to show the left and the right sides of the durations of this peak. And here they are shown at the top of the screen. I can select them to get a sense of the data, see how long they are. And I can already see that my symmetry is going to be below 50% just based on a quick visual examination. So that's one thing that's good with this modern software application. You get that quick visual. For good measure, let's zoom in on this area to get a closer look and do a more visual inspection and validate those intersection points with those capsules. So here is where the left side starts, right at that intersection of that 10% value, and it goes right to where we calculate the maximum of the peak there. And then going over to the right, we see that we want to end at that other 10% intersection at the right-hand side. So in step 9, let's see the result of this calculation. And we see it in the form of the bar chart. And I can get a quick sanity check by looking at the durations of my left and right side capsules in the capsule pane down here in the bottom right. You can see those values. And lastly, let's apply this calculation to a wider range of signals. So here, all our analysis gets applied to the signal throughout the investigate range. One of Seeq's mottos is nail it, then scale it, and it works well when you analyze a lot of data. You hone it in on one particular thing, and then you can easily expand your analysis by just expanding your time range and get all those values. So here, Bristol Myers Squibb was able to take their scientific analysis, deploy it across their signals, analyze for proper quality, and jettison the paper printouts. Their scientists can now spend that valuable time doing experiments or looking for other ways to improve their business. Okay, so that was it for the improved product quality use case. And now I'll move on to the enable continuous manufacturing use case. And this one comes from Just Biotherapeutics. They provided a step-by-step analysis to get insight into their downstream chromatography runs from a Pal Bio SMB device, and it's pictured there in the bottom right. They wanted to use advanced pattern recognition and visualization to get insight into their process. And as we look at this data, it is important to note that there are 3 columns running in different phases. It is important to make sure that they have a consistent and similar performance across all those columns. And in the Seeq application, I will demonstrate how to create a visual output to support this and support their investigation. And the business impact is the optimized purification, leading to greater control of product quality and quantity. So let's take a look at this one. So let's start off by opening the workbook, Enable Continuous Manufacturing. And the output result shown here is a scatter plot. And this is one of the various views that Seeq has to provide that visual feedback to scientists. In this case, the users wanted to see a plot outside of the time domain and rather see it in terms of how two signals are correlated to one another. In this case, the conductivity versus the column volume. So let's start through this, start with the blank worksheet, of course, and next I'll add the conductivity and pump set point signals. And here we can easily see the cycles over the three columns during the run, shown on the same signal here. So we combined all three columns into one place to make it easier to analyze, as you'll see shortly. Because here in step 3, I'm able to create capsules using a value search. I named these purification runs. And here it was just when the pump had a set point greater than 0, and I can highlight them, cycle through them. So all three columns in one place, the same signal. And then next, I can calculate the amount of volume that has flowed through each cycle. We do this by using an integral formula on the pump set point, which is you get a linear signal from doing that since it's a straight line. And then in addition, we also divided the integral by the column volume because we knew the column volume that went through the columns, known quantity, and showed it on that bottom line. And then lastly, we can get a plot like this where we see the conductivity versus the column volume. By adding those signals to the scatter plot, I can click through them and highlight each one of interest, and I see I still have the time domain view and above the investigation bar. And so the last one looks like a bit of an outlier, so I'll leave it highlighted here, and you can hover over it to get values from the scatter plot. Perhaps there's a column volume inconsistency that needs to be investigated here since it differs from those other two curves, hypothetically. So Just Biotherapeutics used this methodology in their R&D process to get overlays and calculated results to improve their manufacturing throughput. All this was made possible by intuitive visual patterns and analytics. And to reiterate the fundamentals of how Seeq allows users to lift that insight from their time series data, here's how we apply capsules in general. As was just the case with Just Biotherapeutics, we laid down these capsules, which can be done in innumerable ways, and we aggregated the values out of those capsules to get the signal that we want. In the previous use case, it was column volume. So regardless of the particular application, the process can be reduced down to this, teasing out the signal in the noise using your engineering and scientific insight. For our final use case, I will turn it back over to Lisa. It's all yours, Lisa.
All right, thanks, Brian. Okay, so in addition to the individual data analytics problem statements that we walked through, we also want, back to the people side, a data analytics strategy that positively impacts our collaboration workflow. So access and collaboration are these key elements, and I'm going to spend a couple minutes on the next slide with this case study, explain the concept for how to use the same framework in a little different way. So back to our current reality, we have manual, time-consuming, individual-owned files, right? The little person there in the bottom who's trying to figure out where something's stored and who did what. And it's not conducive to broad investigation or easily answering FDA queries. In this case, maybe about a product or a set of experimental batches or runs and the conclusions. So wouldn't it be helpful if we could search by a single experiment and see what you learned by yourself earlier or someone else on your team? How about an easy way to pick up on an analysis that you or someone else started and then easily overlay new data, whether it's from that molecule or another one, to look at trends? So in the next clip, we have some screenshots. So this is the same Seeq framework that we've been talking about, but in this case, we can do a little twist where we customize user access here, the user interface, to be able to compare several individual variables across runs, batches, or experiments. So I say it that way because whether it's an R&D lab or a set of ICH batches or a manufacturing organization, we want to be able to view, let's say, the productivity or key ratios of metabolites or crystallization parameter across every experiment done over some time period. And as Brian showed, you have the ability to move back and forth quite easily once you have your analysis in place. So one way to think about Seeq, in addition to the individual problem statements, is to look at how you could go in this case, select across a number of conditions or experiments a key variable. So this gives us an opportunity to capture relationships across a portfolio. It could also be a way to have a little more effective knowledge capture and sharing across teams. So someone isn't waiting for the Excel sheet with the analytics data. They can go in and pull that analytics data to go with their process data. And so I just wanted to make sure we highlighted that there's a lot of different opportunities to leverage across all the case studies we've shown today how this can fit in an organization. And so with that, I'd like to move us to our summary. We know that we can't scale by multiplying computers for each piece of data we want to have. So, let's review our data analytics strategies for extracting the most value. So with the right data historians and databases in place, as shown in the upper left and as we discussed earlier, we can utilize Seeq to overlay the sources for the access, analytics, and visualization needed to gain the knowledge that we want to have from our data. And this next slide shows that one way to utilize the speed of thought workflow, in a sense, within our data analytics framework begins with a question or a need. Whether it's the person needing to respond to an FDA query or it's a senior leader trying to decide what resources to put on which part of the portfolio, we can leverage this framework of having key data sources defined and know where they are with our Seeq analytics layer to do the investigation, the collaboration, monitoring, publishing, exporting, everything we need to do to really get value out of what we have in our data. The framework we've discussed today on the next slide shows how we can do the complete data analytics experience with this framework in place, starting with our business driver, easily moving through how we deal with our data and the investigation, all the way to where we can have some tools in place because this framework with Seeq sitting there on top lets us update in real time new information, new data. There's an opportunity for publishing and monitoring. So putting these learnings into play as opposed to having them stuck in a spreadsheet somewhere or in a drawer in a file. And so we covered a number of case studies today illustrating the broad applicability of this approach. It's important, as we said, to begin by identifying the business needs and then choosing a strategy that enables each of your team members so they can see immediate value. Not an intensive IT project that's going to take a year before the scientist or engineer gets something, but putting something in place where they can start making good progress today and then partnering with IT and others to move this forward. And so the question for you today is, as we wrap up, what steps are you going to take to adopt a data strategy that effectively addresses the business needs of your organization? And with that, we are happy to answer questions. Thank you.
Thank you very much, Lisa and Brian, for that presentation. We are now moving to the audience Q&A portion of today's webinar, and I would like to invite our audience members to continue sending in their questions. And the first question I would like to start with is, Lisa, this question is for you, and the question is, can we use this approach at both R&D and manufacturing scales, and how does it support off-site visibility? For example, I want to analyze data being gathered at a facility overseas or my own facilities while on the road.
Oh, okay. Thanks, Tian. Thank you very much for that question. The short answer is yes. From the standpoint of the way this framework is put together is it offers flexibility for each of those needs. And so, for example, as long as that data is accessible, whether it's an R&D analysis or it's something to monitor batches, it's simply a matter of how we choose to use the Seeq workbench. As far as being able to visualize what's happening, let's say at a site overseas, to me that's where it's very powerful, because you have a chance to get on the phone with somebody who's actually at the site. You can each pull up the relevant time series and contextual data that's happening right then and trend it and investigate it, make comments. And so in many respects, it gives a real-time grab onto what might be going on that also keeps that person from having to maybe get on an airplane to fly over there. So can definitely fit that need.
Thank you very much, Lisa, for that. And the second question I have here is targeted to Brian. And Brian, how does Seeq interface with the data, especially if my data sources are segregated?
Yeah, good question. So what the Seeq server does is connects to your data. We've described that, and we have a variety of connectors. So for process data, we have connectors to all the major process historians, OSI/SoftPy, Delta V, and others. And then for the offline analytics instruments, oftentimes they have historical data stores as well, like OPC, HDA, for instance. We can connect to that. And then also, if they're more custom than that, they don't follow any open standard per se, we have a variety of connectors to read that data right off the machine in text format, so forth. So it's pretty straightforward, easy to deploy from an IT perspective, and just essentially connect out to all those data sources. And then the visual is easy. We just wrap it up into that display window that you've seen, all in one place.
Great. Thank you very much for that, Brian. And the next question we have here is from an audience member, and the question is, you mentioned compatibility with MATLAB, Python, and Java. Could you describe that synergy?
Yeah, I'll take that one, Lisa. You want?
Great.
Yeah, so we have a variety. So let me back up one sec. So all of the commands to the Seeq server, all of the anything that you've seen that I was done with the clicking on the interface, and even more than that, can be done programmatically using a REST API that Seeq has. And the REST API is very flexible and can integrate with any software solution that can be programmed talking to a REST API. And what we did is we took the next step to create some software development kits around the API, so that for users of MATLAB, Python, Java, and C#, they can use those SDKs to basically create some out-of-the-box queries and import data and export data right out of Seeq using those tools that they're used to. So if you want to see a signal, you want to perform math on it, you use the SDK, pull it out, do it in MATLAB, then you can push it back to Seeq, and the possibilities are endless. Just however you want to use your data, the most effective what you're using, you can do it that way with whatever tools we have available there. And like I said, we have all the big ones available.
Great, thank you for that, Brian. And the next question we have here is, can you give a few other example pharma processes where the Seeq application could be a useful data analytic tool?
Brian, I'll take that one.
Yeah, great.
Okay, so we talked a little bit today about chromatography and purification, filtration, bioproduction, but effectively it's any process in pharma that has the opportunity to leverage the time series data about that process. So it could be a tablet coder, for example, with spray rates and temperatures with coding quality attributes. It could be a API crystallization process where we want to look at the robust, how reproducible is the temperature cooling curve and its impact on crystal size and shape and distribution. So really it's where there's a process that has quality attributes that you want to look at in context of the time series data. Thank you.
Thank you very much, Lisa and Brian, for those answers. We have reached the end of the question and answer portion of this webinar, and if you have further questions, please direct them to the email addresses showing on your screen. That's alchemy@alchemyinnovation.com and info@seeq.com. Now, please join me in thanking both our speakers today, Dr. Lisa Graham and Brian Crandall. Thank you both.
Thank you, Dion. Thank you, everybody, for your time today. Appreciate it.
It was my pleasure. Thank you, Deanne, and everyone else who attended.
Thank you everyone else for participating in today's conference. You will be receiving a follow-up email with access to the recorded archive for this event, and a survey window will be popping up on your screen, and your participation is appreciated as it will help us improve our future webinars. We hope that you found this conference informative. Have a great day, everyone.